Hello students, in today's class we'll be learning about ion channels. Most channel proteins in the plasma membrane of animal and plant cells connect the cytosol to the cell exterior and necessarily have narrow, highly selective pores. These proteins are concerned specifically with inorganic ion transport and so are referred to as ion channels. Channels cannot be coupled to an energy source to carry out active transport, so the transport they mediate is always passive, which is downhill. Thus, the function of ion channels is to allow specific inorganic ions, mainly sodium, potassium, calcium, and chloride ions, to diffuse rapidly down their electrochemical gradients across the lipid bilayer. The ionic gradients and electric potential across the plasma membrane, along with the ability to control ion fluxes, play crucial roles in many biological processes. Like all channels, ion channels alternate between open and closed states. When their opening and closing are not affected by the membrane potential or any small signaling molecule, they are called non-gated channels. Whereas the gated channels, like those in the nerve cells, open only in response to a specific ligand or to changes in the membrane potential. Nerve cells in particular have made a specialty of using ion channels. They utilize a diversity of such channels for receiving, conducting, and transmitting signals. Now, let's look at non-gated ion channels and resting membrane potential. In addition to ATP-powered pumps, which transport ions against their concentration gradients, the plasma membrane contains channel proteins that allow the principal cellular ions like sodium, potassium, calcium, and chloride ions to move through them at different rates down the electrochemical gradients. Ion concentration gradients generated by pumps and selective movements of ions through channels constitute the principal mechanism by which a difference in voltage or electric potential is generated across the plasma membrane. In other words, ATP-powered pumps generate differences in ion concentrations across the plasma membrane, while ion channels utilize these concentration gradients to generate a tightly controlled electric potential across the membrane. The plasma membrane of animal cells contain many open potassium channels, but few open sodium, calcium, and chloride channels. As a result, the major ionic movement across the plasma membrane is that of potassium ions from the inside outward, powered by the potassium ion concentration gradient, leaving an excess of negative charge on the cytosolic face of the plasma membrane. This creates an excess of positive charge on the exoplasmic face. This outward flow of potassium ions through these channels, called resting potassium channels, is the major factor for the inside negative membrane potential. In all cells, an inside negative electric potential, or voltage of about 70 millivolts, exists across the plasma membrane of all cells. This is the resting membrane potential. Now let's study the potassium and sodium channels. Perhaps the most common ion channels are those that are permeable mainly to potassium ions. These channels are found in the plasma membrane of almost all animal cells. An important subset of potassium ion channels are open even in an unstimulated or resting cell and are hence sometimes called potassium ion leak channels. Although this term covers a variety of different potassium channels depending on the cell type, they serve a common function. That is, by making the plasma membrane much more permeable to potassium ions than to other ions, they play a critical part in maintaining the membrane potential or voltage difference that is present across all plasma membranes. All ion channels exhibit specificity for particular ions. Potassium ion channels allow only potassium ions, but not even closely related sodium ions to enter. Likewise, sodium ion channels admit only sodium ions, but not potassium ions. Comparisons of structures and sequences of potassium channels of diverse organisms like bacteria, fungi, and humans established that they all share a common structure and seem to have evolved from a single type of channel protein. Now let's study the structure of the resting potassium channel. All potassium channel proteins are tetramers comprising of 4 identical subunits, each containing 2 conserved membrane-spanning alpha helices called S5 and S6 by convention, and a shorter P or pore segment. The non-helical P segments are located near the exoplasmic phase and connects the S5 and S6 alpha helices. Owing to its structure, the P segment lines the ion pore and forms the ion selectivity filter. And it is this filter that allows only potassium ions and not other ions to pass. Now let's study gated ion channels. The main types of stimuli that are known to cause ion channels to open are a change in the voltage or membrane potential across the membrane, and we call them voltage-gated channels, a mechanical stress in mechanically gated channels, or the binding of a ligand in case of ligand-gated channels. The ligand can be either an extracellular mediator, specifically a neurotransmitter in case of transmitter-gated channels, or an intracellular mediator such as an ion in case of ion-gated channels, or a nucleotide in nucleotide-gated channels. The activity of many ion channels is regulated in addition by protein phosphorylation and dephosphorylation. Now let's come to voltage-gated cation channels. The fundamental task of the neuron is to receive, conduct, and transmit signals. The plasma membrane of all electrically excitable cells, not only neurons but also muscle, endocrine, and egg cells, contains voltage-gated cation channels, which are responsible for generating the action potentials. An action potential is triggered by a depolarization of the plasma membrane, that is, by a shift in the membrane potential to a less negative value. It is followed by rapid repolarization. An action potential results from the sequential opening and closing of voltage-gated potassium and sodium ion channels in the neuronal plasma membrane. It then spreads as a traveling wave from the initial site of depolarization to involve the entire plasma membrane. Let's look at the operational model of the voltage-gated sodium ion channel. Let's look at the operational model of the voltage-gated sodium ion channel. As in the non-gated potassium ion channel, 4 transmembrane domains in the protein contribute to the central pore through which the ions move. In step 1, in the closed resting state, the voltage-sensing alpha helices, which have positively charged side chains, are attracted to the negative charges on the cytosolic side of the resting membrane. This keeps the gate segment near the cytosolic phase in a closed position that blocks the channel, preventing entry of sodium ions. Step 2. In response to a small depolarization, the voltage-sensing helices move through the phospholipid bilayer towards the outer membrane surface. This causes an immediate conformational change in the gate of the cytosolic phase of the protein that opens the channel. Step 3. Within a fraction of a millisecond, the channel inactivating segment moves into the open channel, preventing passage of further ions. Step 4. Once the membrane is repolarized, the voltage-sensing helices return to the resting position. The channel inactivating segment is displaced from the channel opening and the gate closes. The protein reverts to closed resting state and can be opened again by depolarization. In addition to the inactivation of sodium ion channels, in many nerve cells, a second mechanism operates to help bring the activated plasma membrane more rapidly back towards its original negative potential, ready to transmit a second impulse. Voltage-gated potassium ion channels open so that the transient influx of sodium ions is rapidly overwhelmed by an efflux of potassium ions, which quickly drives the membrane back towards the potassium ion equilibrium potential, even before the inactivation of the sodium ion channels is complete. These potassium ion channels respond to changes in membrane potential in much the same way as the sodium ion channels do, but with slightly slower kinetics. For this reason, they are sometimes called delayed potassium ion channels. Sodium ion channels are not the only kind of voltage-gated cation channel that can generate an action potential. The action potentials in some muscle, egg, and endocrine cells, for example, depend on voltage-gated calcium ion channels rather than on sodium ion channels. The amino acid sequences of the known voltage-gated sodium, potassium, and calcium ion channels show striking similarities, suggesting that they all belong to a large superfamily of evolutionary and structurally related proteins. Now, let's come to transmitter-gated ion channels. Neuronal signals are transmitted from cell to cell at specialized sites of contact known as synapses. Transmitter-gated ion channels are specialized for rapidly converting extracellular chemical signals into electrical signals at these chemical synapses. The channels are concentrated in the plasma membrane of the postsynaptic cell in the region of the synapse and open transiently in response to the binding of neurotransmitter molecules, thereby producing a brief permeability change in the membrane. The best studied example of a transmitter-gated ion channel is the acetylcholine receptor of skeletal muscle cells. This channel is opened transiently by acetylcholine released from the nerve terminal at a neuromuscular junction, which is a specialized chemical synapse between a motor neuron and a skeletal muscle cell. The acetylcholine receptor of skeletal muscle is composed of 5 transmembrane polypeptides. When 2 acetylcholine molecules bind to the pentameric complex, they induce a conformational change that opens a channel. The channel remains open for about 1 millisecond and then closes, like that of the voltage-gated sodium ion channel. The open form of the acetylcholine receptor channel is short-lived and quickly flips to a closed state of lower free energy. Subsequently, the acetylcholine molecules dissociate from the receptor and are hydrolyzed by a specific enzyme, acetylcholinesterase, located in the neuromuscular junction. Once freed of its bound neurotransmitter, the acetylcholine receptor reverts to its initial resting state. Neuromuscular transmission involves the sequential activation of 5 different sets of ion channels. Transmitter-gated ion channels differ from one another in several important ways. First, as receptors, they have a highly selective binding site for the neurotransmitter that is released from the presynaptic nerve terminal. Second, as channels, they are selective as to the type of ions that they let pass across the plasma membrane. This determines the nature of the postsynaptic response. Excitatory neurotransmitters such as acetylcholine, glutamate, and serotonin open cation channels and cause an influx of sodium ions that depolarizes the postsynaptic membrane towards the threshold potential for firing and action potential. inhibitory neurotransmitters such as G-aminobutyrate acid, we call GABA, and glycine, by contrast, open chloride channels. And this suppresses firing by keeping the postsynaptic membrane polarized. Let's conclude with these following points. Channel proteins form aqueous pores across the lipid bilayer and allow inorganic ions of appropriate size and charge to cross the membrane down their electrochemical gradients at rates that are about 1,000 times greater than those achieved by any known carrier protein. So the function of ion channels is to allow specific inorganic ions, mainly sodium, potassium, calcium, or chloride ions, to diffuse rapidly down their electrochemical gradients across a lipid bilayer. These ion channels are called non-gated ion channels when their opening and closing are not affected by the membrane potential or any small signaling molecule. On the other hand, gated ion channels are those which usually open transiently in response to a specific disturbance in the membrane, such as a change in membrane potential, as in voltage-gated channels, or the binding of a neurotransmitter, as in transmitter-gated channels. Ion channels are ion selective. Potassium ion channels allow potassium ions only, but not closely related sodium ions to enter, whereas sodium ion channels admit sodium ions but not potassium ions. Potassium ion channels play an important part in determining the resting membrane potential across the plasma membrane in most animal cells. Voltage-gated cation channels fluctuate between open and closed states and are responsible for the generation of self-amplifying action potentials in electrically excitable cells such as neurons and skeletal muscle cells. Transmitter-gated ion channels convert chemical signals to electrical signals at chemical synapses. Excitatory neurotransmitters such as acetylcholine and glutamate open transmitter-gated cation channels and thereby depolarize the postsynaptic membrane towards the threshold potential for firing and action potential. While inhibitory neurotransmitters such as GABA and glycine open transmitter-gated chloride channels and thereby suppress firing by keeping the postsynaptic membrane polarized. So, ion channels work together in complex ways to control the behavior of electrically excitable cells.